Thanks, Peter. Hello, everybody, and welcome to Accelerate Genomics Discovery with SRA in the Cloud. As Peter said, my name is Yuri, and I am the SRA product owner. However, before we get started with the webinar, I would want for you to take this poll. This can tell me what your interests are in this webinar, and I can tailor the message in the webinar based on the results here to make sure that you get the most benefit out of this webinar. And this is an interactive poll, so just click on your screen. In a couple more seconds, when everybody finishes answering, I'll share the results with everybody, and these are anonymized results, so we'll just see the percentage for the options that got picked. Great. The major interest here is why should you use SRA data in the cloud, and also what technologies you should know about if you're going to be doing work in the cloud. And the third topic of interest is you're just here to learn about cloud data and how to use cloud. So in this webinar, we'll cover some basics like introduction to SRA and why SRA was moved to the cloud, the benefits that you'll get from working in the cloud, and of course, the tools we've made available so far. I'll cover 2 case studies that highlight cloud usage and provide you some information and links of where you can go to get started with using cloud resources. And at the end, we'll hold the Q&A session. So for those of you who don't know about SRA, we are the Sequence Read Archive from NCBI. We started in 2008, and our primary mission is to store and distribute next-generation sequencing data. We accept all types of samples and all types of data from all types of sequencing platforms. And this data is used in a variety of different ways, things from bioinformatics methods development to detection of novel genes or microbial or viral species even. So why did we move to the cloud? Why did we do it 12 years after we started? Well, SRA has been growing exponentially year to year. And at this point, we are over 14 petabytes of data. And we have over 25 petabytes of user-submitted files that we can share with the scientific community. And this is a lot of data. And moving it from Bethesda in Maryland to anywhere in the world is a very technically complex task and also a very time-consuming task. Some of our users need to wait weeks or months to just download data before they can even do any computational analysis on it. And of course, sometimes they discover that some of the data they downloaded is not useful and they need to download more. So this is a really big barrier to doing quick analysis on the data that's available. And because there's such great benefit from it, we want to speed this up for the scientific community. And so we went ahead and moved all 14 petabytes of SRA archival data into the cloud. And from there, you can get faster access to this data. You have powerful compute resources in the cloud that are available to you on demand. You can easily expand the storage that you are using in the cloud, so you're never waiting to get more storage. And the cloud can be a great collaboration platform. You can have your collaborators who might be in different parts of the world log into the same system, run the same pipelines, and look at the same results whenever they want. You don't need to share those results individually anymore, since everybody can log into the same server. What we've done so far is, as I mentioned, is we moved all of the SRA archival format to the cloud, and that's available now. We also started moving the user-submitted files, which is roughly 25 petabytes more of data. And this is a new feature from SRA. So instead of using our archival format, you can get the files that we received from the submitters. And as we're always trying to be more efficient, and we want you to be more efficient in the cloud, we started producing contigs ourselves. For now, we only produce contigs for metagenomic data sets, but we will certainly be branching out to RNA-seq, genomic data sets, and other type of data sets as time goes on. Now, in order to access all of this data, we've updated our API, which is the SRA Toolkit, to work in the cloud as well. So instead of downloading data from Bethesda, you will be accessing the data in our cloud buckets if you're working in the cloud. So this will speed up file access. Another new feature is we've made available our metadata in native cloud architecture. And for now, this is just Google's BigQuery product, but this was a very popular feature, and we will be moving all of this also to Amazon's Athena, now that we've received positive feedback from the scientific community for this. So the two case studies I wanted to highlight was the NCBI virus discovery Codeathon, because I think it's a good example of cloud computing and the benefits of cloud computing, and our interaction with a cloud customer who wanted to do things natively in the cloud. So the NCBI virus discovery Codeathon was started with the assumption that there's a lot of viral data available in metagenomic datasets. And so we brought several teams together, and the goal was to build some analysis and processing pipelines for this data set. Now, I won't be going into the details of the actual Codeathon or the results of it, but if you are interested to read about it, there's a publication link you can follow. But what I want to talk about is the pre-computation that had to happen before we can hold the Codeathon. The assumption was that everybody would be working on contigs and not raw data. So what SRA did was we took 140,000 runs of metagenomic datasets that were 7 gigabytes or smaller in size, and we built contigs from this. And so this was either de novo assembly or guided assemblies. And we ended up building 3.9 billion contigs out of all of this data. And in order to accomplish this, we actually had to take a two-step approach in our computation. The first one was using 500 nodes, or like 500 servers, machines, that were only 2 cores and 8 gigabytes in memory. These are very small machines. I think some of our cell phones are more powerful than this at this time. And of course, we had to use larger machines for the bigger files. And for those, we used 100 machines, but these had 96 cores and 300 gigabytes of RAM. So these are big, powerful machines. Well, why did we do it in the cloud? Well, it only took us a couple of weeks to do this in the cloud, but we knew that if we tried to do this internally at NCBI on our high computing cluster, it would take us several months to do this. So you can see the large change there from a couple of weeks to several months of computing all of these contigs. And that's because SRA is not the only resource at NCBI. We share that high computing cluster with other resources. And of course, we would be queued up for all of our computational needs. And this is something that some of you might have faced before as well at your universities. However, in the cloud, all of the compute is what we want, and it's at our fingertips here. So what we learned from this experience is if you want to get started faster in the cloud, collaborations help with this. So if you're collaborating with bioinformaticians or programmers who are familiar with cloud architecture, they can help you get started faster. But you can certainly do this on your own. But if you're going to do this on your own, make sure you start working in a small scale first. This will help you learn the different technologies. And if you're using known data sets that are small and quick to compute on, you know what the results should be. And as you get more comfortable with technology, expand slowly. Premature scaling to 100 or several hundred machines can lead to a large problem, and you won't know where to debug the problem. So it's very important to scale slowly so you can understand what's happening. And if you run into any issues, you can understand where to look for the problems. Now, if you want to know what technologies we used in our computation approach, that was Kubernetes, Docker images for the tools. There was some scripting using Python and Bash and SnakeMake. So these are not the only cloud technologies available. They obviously have competitors. And so as you're looking into cloud technologies, some of those other technologies might be a better fit for you than the ones we used. And we tried to make our compute cheaper, so we used preemptible or spot instances, depending on which cloud vendor you're going to use. Those are cheaper options for compute that you should take a look into. On the notes section of this presentation, I added links to different resources that you should check out. So after the webinar, feel free to look at those to get more information about any of these different topics. The community use case that I wanted to highlight was from a researcher who was interested in the evolutionary relationship of microbial communities in marine environments. So they were looking at marine metagenomic data sets. What they really wanted to do was build up an automatic pipeline to look through SRA data, and the pipeline would run in the cloud. They need to be able to search for these data sets quickly and efficiently, and they wanted to be able to fine-tune their search parameters for these data sets very easily. And their other need was to be able to actually know what the microbial communities are within the SRA data sets. So for this, we thought that BigQuery or Amazon's Athena products would be a good fit. So we started doing our testing in BigQuery. And what we started out with is putting SRA metadata there. So we put sample information, so where the sample was collected from, like what depths, all of this metadata that users provide for us. We also provided sequencing information of how the sample was sequenced in terms of what platforms were used. And also some basic statistic information, such as the number of bytes or the number of reads or number of bases available in this data set. So this allowed the user to select high quality data sets from a particular sequencing platform they were interested in, or if they wanted the run to be of a certain size. Now for their second need, which was to know the taxonomic content of the actual SRA run, for that we had to make the results from the SRA taxonomic analysis tool available in BigQuery. Now we haven't released this publicly yet, as we were still going through testing with a small user group, but we will be releasing this soon, and all of this functionality will be copied to Amazon's Athena as well, given a little bit more time. So with this data set, they can actually identify what microbial communities are available in a particular run so that they can fine-tune their search parameters even more. And since this information is available very quickly from BigQuery, what they can do is take the search based on sample and sequencing information, take the search results from the taxonomic content, and combine them and kind of take the intersection of them, thereby getting a very small data set of runs that they can run more expensive computational analysis on. So getting started is actually fairly easy. What you will get out of this will be very customized filtering for data sets that you want to analyze. You will obviously have very powerful compute available to you, and all you need is a laptop to log into the cloud architecture. If you're worried about costs, both Amazon and Google have free tiers available where you can start playing around with this new technology and moving your analysis pipelines or processes into the cloud. And if you're working with small data sets on small machines, all of this can be free for you. So definitely don't be worried about it. And if you need to actually get the data out of the cloud, that's called egress. And you should really only be egressing things that are the results of your analysis. That is much smaller in size than actual raw data or any kind of middle files that you might generate between different tools. And if you're ready to get started there, we've made videos available on how to start a cloud instance in both Amazon and Google, so you can check out our YouTube channel for that. We've also provided help documentation for all of this information in terms of getting started with computing and some example queries for BigQuery. So you can find all of this in our help documentation. Now that I introduced the tools we've made available to you, we are always looking for more feedback from the scientific community. So please reach out to us. Tell us if something works for you or something doesn't work for you. That's even more important. because if we know what's not working, we can try and fix it and make the tools better for you. So the more feedback we get on anything, either for SRA or NCBI, will pay dividends to you back as we can improve our resources to meet your needs. So if you want to just continue following NCBI and what we're releasing into the cloud, you can follow the NCBI Insights blog, where we'll be posting more information about upcoming new functionality. We also send out messages on Twitter, and you can follow NCBI for that. SRA is also registered with the Amazon Open Data Registry, so you can find us there as well. And that's about it. So I can now open the floor to any questions that you guys might have.
Hi, Yuri, this is Peter. So we have some questions in the chat pod. And then I think I'm going to ask you, you were thinking about doing a demonstration of BigQuery. If we have time, I think we can do that because somebody did ask that one.
Okay.
But there's some other simpler ones. Somebody asked a question about sequences that are submitted to ENA, to the European Nucleotide Archive. So I think we basically get those sequences into SRA, and those would wind up on the cloud. Is that not true?
That is true. Since SRA is part of the INDC, everything that is part of SRA is also available in the cloud as well.
Okay, another one that's kind of similar in spirit. They were asking whether SRA is mirrored or split between AWS and GCP, and I believe it's mirrored in both places, so you can get it on either platform. Is that correct?
That's correct. It's perfectly mirrored, so you can, depending on your needs, you can use either platform.
And one other simpler one, although it's kind of a sensitive topic because we don't like to talk directly about the costs that we've experienced from the government point of view, but can you comment on the compute costs associated with config creation?
So that is definitely a difficult topic to answer. I don't know the specific number, but it was probably several thousands of dollars or tens of thousands of dollars in terms of computation. I don't know how that would relate to anybody else's expenses, but considering the amount of data we processed and how fast we processed it, I think it was a good expense for us.
Okay, thanks, Yuri. And so I guess the other question was, somebody was asking about using BigQuery and the metadata, and so you had a demo that you could do with that. That might be useful to do if you have time.
Sure, I can do that. So, of course, the easiest way to find BigQuery is just to do a Google search for it. Once you're here, if you don't have a Google account, you'll probably will need to create it. But from here, it's very simple. You can go to the console page. So this is the web interface that Google provided for their service for BigQuery. And I think it's fairly intuitive to use. So you can see your queue query history, the saved queries you've run, and some other options on here. Now, in order to connect to the SRA data in BigQuery, I find it easier to pin our project. So I'll unpin my project here. I want to go to the Add Data section here, click on it, and there's a pin project selection. So our project is called NIH-SRA-Data Store. So I type that in and just click on the pin button. Let's try that again. I might have had a typo there. Okay, well, I might be typing something there, but once you add the data store, you can take a look at what's available there. But I'll use one of my saved queries here to find some runs for you. So I can just click on Open Query and click Run. So in order to use BigQuery, you'll need to learn SQL or SQL, and we have some example queries available on documentation pages, so you can copy those to get started. There's a lot of very useful tutorials online for using SQL and learning SQL, but to kind of get started, it's not very hard. So I have a more complex query here, but as you saw, I ran the query, the results came back in seconds, and you can see that I selected different type of runs, either from DDBJ for this one was submitted to SRA, and I displayed some columns that might be useful to me, such as the number of spots used in the analysis and percent of unidentified reads. So between the performance of BigQuery and the ability to write your own queries and customize the results, this is a very useful tool for pipeline developers, or even if you just want to get access to this data very quickly, you can use the web interface to search for your data sets. So I think it's a very powerful tool that can be very beneficial.
Thank you, Yuri. We got a couple of more questions. Actually, there's probably three of them or four of them actually, that we should probably address. Someone asks, is there an SRA Docker container store for SRA Toolkit and other apps?
So SRA will be releasing a Docker image with the SRA Toolkit. It's in our roadmap to put that on Docker Hub, so that will definitely be coming soon. NCBI has a Docker Hub account where you can look for other products that NCBI supports in Dockers. So please check out NCBI at the Docker Hub.
Another question that's sort of a general one that's probably useful to address. It has to do with what data are publicly accessible, and I think it's probably worth talking about the difference between the controlled access data and the data that's not controlled access, and what one would have to do to get to those things.
Right. So we have public data that's available right now. For controlled access human data, you would need to get access to that data set through dbGaP first, but that data is available in the cloud as well. So if you have access to data sets through dbGaP, you can certainly access them in the cloud as well.
Okay, thank you, Yuri. Another question had to do with the contigs, and I think you mentioned that they should be available before too long. How soon do you think they will be available?
So the contigs for metagenomic data sets are available now, and I think I added the link to the documentation for them into the presentation, but if it's not there, I'll make sure to send out the link to the documentation for them after the presentation. And the contigs are available in both Amazon and GCP.
So the other question was about the compute cost per assembly, but I think you addressed that when you talked about the cost that we incurred when we were doing those contigs.
Yeah, unfortunately, I can't answer the question of cost per assembly, but I can certainly get in touch with whoever asked that question to talk to you more offline about that topic.
Yeah, that's a good idea. So you can certainly contact, you can write in to the webinars address if you want to, webinars@ncbi.nlm.nih.gov. And someone also asked, can you show an example of actually doing an assembly in the cloud for a particular SRR?
So doing assembly in the cloud is not any different than running assembly at your local server. You use the same tools as you would there as well, and the only difference is that instead of running it at a local server that you have available to you or your desktop, you're doing it on a server that's located in the cloud. But the actual process itself is not any different. So in this case, I would just be calling up an assembler and providing data to it. I don't have a demo available right this minute, but I'm sure that SRA can make a demo of that and share it.
Okay, another question, which is, I think you addressed this, and in some ways this webinar was about this very topic, but it's probably worth just reiterating. So the question is, what is the advantage to use EC2 instance instead of using my own computer, which is basically using the cloud instead of this person's own computer? So maybe there's just a couple of points to sort of finish things off.
So if you're downloading data using the SRA Toolkit, it usually pulls data from our servers, but that's also where everybody around the world is accessing the data as well. So you're obviously competing for resources there. In the cloud, it scales basically automatically. You get faster access to the data there as well. Also, if you need more powerful compute that's not available locally to you, you have those machines available in the cloud as well. So as I said, we had those bigger machines that had 96 cores and 300 gigabytes of RAM. So those might be harder to get locally for you, but they are available to you easily on the cloud architecture. So those are kind of the benefits of doing that, where you get faster access to data, more powerful computes. So if you move your pipelines and processes there, you can see a speed up in the overall analysis.
Thanks, Yuri. That was a good question, and that's a good way to sort of seal things when we're talking about this, the main advantages of doing this. We mentioned the access to the contigs, which are now available. Is it easy for you to show us how to get access to them?
Sure. So here I'm just going to the virus discovery GitHub page, and there's a README on how to get access, and we provide instructions of how you would want to download the config. So in this case, if you're somewhere in the cloud, either Google or Amazon, you can run either this AWS S3 copy command and download the config file to your processing area in Amazon. There's a similar command with GSUtils for Google. You can even run a wget command from a cloud instance running in Amazon to download the data. So that's all it really takes, is running this one command to download one of the configs FASTA files.
Okay, that's great. I don't see any more questions. I think we're just about done. Oh, wait, somebody says they have one more in just a second. Okay, let's see what this one is. So this is an interesting one. We might not be able to address this directly, but this is about the data from GISAID, and how does that data get reflected in SRA? This is particularly relevant for the coronavirus data.
Right. Unfortunately, I don't believe we have that data available at SRA. We are certainly exploring options on how to get access to that data, so it would be publicly available.
Okay, Yuri, I think that's all the questions we have. And if there's any that we didn't get to, we'll be sure to address them in the document that we're going to produce. So I think we can go ahead and wrap this up. Thanks everybody for coming. And thank you, Yuri.
Thank you.